Good morning, sir.
Yes, sir.
Sir, should we start?
Good morning.
Sir, should we start?
Yes, kindly start.
Okay. A very good morning to you all, and
Yes.
happy Vijaya greetings to you all.
Yes, start. Kindly start.
So welcome. I would like to welcome you in the next generation big data biology. This 5-day long workshop. Actually, in microbiology, people were using single microbes. They used to isolate single microbe from soil, water, and whatever the source may be, and cultivate the thing. But like Human Genome Project, we are now focusing our experiment, our research on bulk microbiota, maybe from desert microbiota, from water microbiota, or from our gut microbiota. We know gut microbiota, they are responsible for different things. They can guide our thinking, our personality even. So let us start the research on this microbiota. And for analyzing this microbiota, this microbiome, we have to use metagenomic analysis and big data analysis. For this, we have to prepare, we have to know how to prepare this library of this genomic sequences. And for this reason, we are going to arrange this 5-day long workshop on this big data analysis. And regarding from the standpoints of the students, we are also the student of biotechnology, and we will be the co-learner of this project, learner of this workshop. So please be with us. And now I would like to invite our Honorable Vice Chancellor, sir, to address to officially inaugurate this workshop. So. So kindly inaugurate this workshop.
Okay, thank you. Can you hear me?
Yes, sir.
Yes, I am facing some connectivity issues. So maybe this internet connectivity get disrupted several times. And without wasting time, let me start my deliberation. It's very good that we are organizing one of the important area webinars on an important area in big data analytics in biology, and that's too in a time of festive season when people are on festive mode. So it indicates that we have become this little bit professionals in our approaches in imparting teaching, learning, educations, and knowledge sharing with the learning community. So this is a very for the university. Next, regarding this topic matter, aspects of this big panel, and will be very sure get transformed into nanobiology, because this emergence of this researches in nano areas in biological sectors and micro dimensions in other areas also that are going to lose its relevance. People are working on this dimensions. And the important area is biology has become in the biological research only, but it is propagating and percolating and penetrating in other areas too. And that we are observing in mitigating food problems, in mitigate different types of diseases and all these things. There are lots of researches, even in different other areas like robotics and other areas. Then the mimicry of biological systems that are taking this front seat. So that's why this importance in biological research, and that's true in microbiological research. Nanobiological research is getting paramount importance in the current context and in the future scenario, future this world also. And big data now, it is very important because now people are working with, people have their tools and technologies to work with, these different types of data. So this variety, veracity, these different, these dimensions of data now can be taken into considerations. And earlier it was not possible. So we had to, this get restricted in researches with a small amount of data. Now, as IT information technology has proliferated to a very significant extent, and handling of this, this big volume of data is not so problem. Therefore, the translation of the research into practical, things, scenario, or this reality now is this very formidable. So that's why uh, these understandings of these tools and techniques for handling big data is very important. And this understanding of the importance of researches area in the different these facets and different aspects of microbiology is very important. And I believe that this combination will be quite helpful in pursuing research activities, and in transforming our economies also, with the development of some this with the translation of some knowledge into practical reality. And for our country it is very much required. We have enough potential. Now this is the time to this take a this massive integrations of all of our knowledge areas, all our expertise areas, to embrace all these current tools and techniques. And I believe that out of these workshops, the numbers of participants who have joined here, they will be immensely benefited, and they will be inspired. They will be they'll be seeing the problem from different dimensions, for to take that take the problem for a solution or mitigations. And with this, my sincerest thanks to organizer Dr. Sarojit Dash, Dr. Rinarani Rai, and all other faculty members of our School of Biological Science for organizing these relevant programs, timely important programs in this particular time. And my thanks to the resource persons also, because they have consented to spare their times at this time, particular this moment, for the benefit of our learning community. And I believe that it will be a great learning opportunity for all the participants. So with this, I would like to request Professor Rinarani Rai to take this program ahead, and I believe that it will be a great success after these 5 days or 4 days. And kindly take a stock of it, and kindly this arrange next levels of programs in this series, so that after some time,
Sir, please unmute, sir.
or good numbers of words for the benefit of our learning community. Thank you.
Thank you, sir, for your illuminating and enchanting and supporting lecture. So now I would like to request Dr. Sarojit Dash to kindly introduce our resource person and start the technical session. Dr. Sarojit Dash? I think
If connection is an issue with Dr. Sarojit Das, you kindly start this program.
so I'd like to invite Mr. Subhrajit Roy to kindly start the session.
Thank you. So I would like to share my screen first. Please let me know whether you are able to see this. Or
Yeah,
Yes,
we
sir.
can see. Yeah, we can see. Go to the first slide.
okay.
Right. Okay.
Thank you. I would like to provide my sincere regards to Professor Shoikat Moitra, Honorable Vice Chancellor of Maulana Abul Kalam Azad University of Technology, and Dr. Sarojit Dash and Dr. Rina Rani Rai for organizing this wonderful webinar series. And I am Subhrajit Roy, and I will be discussing about the metagenomics. So through my PhD career, I am a neurobiologist. So I came across a certain situation where I realized that the importance of studying microbes for apart from the genetics and the proteomics, what we have been doing for so long, their importance of microorganisms are getting important day by day, and they can influence, as Dr. Rina Rani Roy was saying, that it has great influence on our personality and our moods, and even it has been associated with several disorders. So today I will be discussing on that. So uh, moving on to my talk. So before going into the details of metagenomics, I would like to discuss something about the genomics. For the last 2 decades, if you go back, you will find that in the field of genetics, some monumental achievement has been performed, such as the Human Genome Project, which was initiated in 1990 and was completed in 2003. So this Human Genome Project has not only greatly accelerated our biomedical sciences, but has also transformed it. So the genomics is a subdiscipline of genetics where it is devoted to mapping, DNA sequencing, and functional analysis of the genome. So it is the study of all the genes which are present in an organism. And not only that, it also studies the intergenomic phenomena and other interactions which are associated with the genome. Thomas Roderick in 1986 first coined the term genomics, and it includes 3 phases. First one is structural genomics, which includes the 3D structure of the protein, and we correlate those 3D structures with the genetic sequences. And functional genomics is a more dynamic approach where we deal with translation and transcription, and assign those functions to the gene. Rather, we actually do that the address that what gene performs at RNA and protein level. So this is what we call functional genomics. Comparative genomics is something where we compare the genomic architecture of one particular species to another one. Take for example, if we compare our human genetic sequences to chimpanzees, you will get 83% similarity. That is what we call a comparative genomics. So moving on to the next phase, where we will be talking about the metagenomics. So metagenomics is, before going into that, I would like to tell you that you might have heard of several omics associated terms like transcriptomics, proteomics, metabolomics. So these high throughput studies have helped us to learn how genes function on the protein and RNA levels, and how the system approach is more important rather than studying an individual entity or single event. So the term metagenomic was first coined by Jo Handelsme in 1998. So it is a technique where we isolate the genetic material directly from the environmental sources. So it might be soil, it might be water or fecal matter from any animal, and it is completely different to the single genome approach where we just take one individual or one bacteria, isolate its genome and send it for sequencing. So instead of that, we are collecting studying it as a community instead of studying the individual entity. So it may be referred to as environmental genomics, ecogenomics, or community genomics. So it is very increasingly important day by day because it has helped us to understand the human health medicine. It has great importance in agriculture, energy production, bioremediation, and even it has helps to identify microbial genes and its novel microbial genes and its associated metabolic pathways. So we know that majority of the bacteria which are present in on this earth cannot be cultured because either we don't know its nutrient condition, or we don't know the exact, or it may require the presence of other bacteria for their own survival. So for that reason, metagenomics becomes increasingly important because we don't need to culture them one by one. We can directly extract the genetic material from any environmental sources. What are the microbes will be present there, their genomic material will be collected, and we can send it for high throughput sequencing. Then we will be through computation and analysis, we will be able to identify those genes or assign the genetic architecture or functions depending on what kind of approaches that we are considering for this study. So you might be thinking that the students particularly might be thinking that why should we study microorganism? They are microscopic and hardly negligible. So there are other important things which might be interesting to us rather than studying microbes. So those who have an idea that microorganisms are mostly disgusting and associated with different kinds of disease, trust me, you are wrong. There are many organisms which are present, and I believe that we thrive on this planet because of the microorganisms are there. So they have created an environment. They are always communicating with us through their metabolites, maybe directly, and that's why we are able to survive. So first of all, the nitrogen fixing bacteria which are present on the soil, as you know, the plants are unable to utilize directly nitrogen as its source. So there are nitrogen fixing bacteria present in the soil, and which fixes the aerobic nitrogen into some compounds that becomes accessible to the plants, and they can utilize it for the development, and we get fruits and crops, and become important to our survival. Microbes which are present within us, the gut microbes particularly, they can interact with our immune system and boost up. And not only that, they are associated with several diseases. Even the presence or absence of any particular microbe in our body can be an indication of a disease condition. So that is an important thing. Would like to discuss on that in later phases. And there are certain bacteria which help in bioremediations. One of them is oil-eating bacteria like Pseudomonas aeruginosa. And we often heard of oil spillage, which occurs in the marine environment, and it's very detrimental for the aquatic environment. So if we can employ this kind of oil-eating bacteria, we can eliminate this problem, and this is becoming increasingly important to us. And of course, there are enemies. There are Vibrio cholerae, which is causing cholera, Salmonella typhi, and there are many more in the list. I don't want to name all of them, but you can understand how the microorganisms are influencing our lifestyle day by day. So it is all not only in the presence of food. It provides fermented products to us, and also it's associated with different disease conditions. So why we need to study it as a community? Why we don't need to study it individually? The importance is that I have, as you can see in my slide, I have put a picture of Sachin Tendulkar. So he is one of the finest player we have in our Indian cricket team. So when he was in form, when he was playing at its best, think of a condition he is playing alone. Even if he scores his best of its performances, he cannot win against a team. And on the other hand, when we have an entire team, whatever disproportionate or whatever smallest amount of work that a person in a team is performing, that might be very important. Maybe in a team, one person is saving a boundary, or maybe taking a catch. That will become a most important criteria for winning a match, right? So same thing for the microbial community, that there might be many bacteria, and the relative abundance of those bacteria might be different in different areas. But as we have, while I was introducing this metagenomics to you, I have told you that we thrive on this planet because of the microbes. So they are intricate part of our system. So the plants, they are animals have always are thriving with commensal or relationship between with the organisms. So they provide nutrients, they provide essential metals and vitamins they make available to us. And of course, through fermented products, we get yogurt from the fermented foods. And there are many in the list, but so the microbes also remediate toxins which are released into the environment. So it's present in our all over our surface, body surface for the humans, even in our skin, mouth, genitalia, gut, all over the places these microbes are present, and always they are influencing and our health. So these functions are conducted in a complex community. So intricate, balanced, and integrated entities that adapt swiftly and flexibly in the environmental changes. Microbial communities often interact with the medium, which whether it is present in soil or water, or where they grow, exchange nutrients, biochemical products, or often they interact with some chemical signals through direct cell-to-cell communication. So this is what we, the microbiologists who have been associated with the pure cultures, know this is a traditional way of doing a microbial genetics, that they isolate a bacteria from the environment. They identify the nutrient conditions which are required for its development. They plate it or grow it in a liquid media, and they isolate the bacteria, the genetic material from the cells and send it for sequencing. That's what they do. That is a traditional way of doing a microbial genetics. But on the other hand, we are considering metagenomics, where we don't do it when isolating cultures. We isolate the genetic material directly from the community, not by segregating one by one. So whatever the microbes, whatever the components will be present in that area or sample, will be isolating the genetic material and send it for high throughput sequencing. So either it can be a survey like 16S rRNA sequencing, or it might be a whole genome sequencing where we are considering the entire genetic material as a whole, not any particular region of that genome. So why genomics is getting more and more important? So why it's the metagenomics getting more and more important? Why genomics is not sufficient for that? Because of the microbial diversity. Even on us, we can have about 100 trillion microbial cells present on all over our body. When there are limitless microbial diversity, consider about a small soil sample, you will get about 40,000 microbial cells in a soil sample with limitless variations in these bacterial strains. So this is beyond the human capability of isolating individually and doing it, doing the genomics, in the traditional way of doing a genomics. And most of the microbes cannot be cultured. You know about Microbacterium leprae, which cannot be cultured, and there are many more. Actually, there are 0.1% which are culturable, and this is because either we don't know the correct culturing conditions of those bacteria or microbes, or that even it is necessary the presence of other bacteria in the vicinity for where they exchange metabolite, nutrients, or chemical signalings are there for its survival. So it is important to isolate the bacteria directly from the environment. And even consider you are isolating a bacteria and providing a particular nutrients for its growth. So what will happen is that there will be disproportionate distribution of bacteria in that soil. Take for example, you are isolating soil bacteria, and when you have provided a particular nutrient for its growth, all of them will not grow equally. So some of them will be in a better position to utilize those nutrients, and will exclude the small or the proportion of bacteria which are present in small amount will be eliminated from the system. And when you sequence them, you will not have any idea whether it is only few bacteria are present there, or there are many which we are unable to detect. So this is a timeline, and how microbiology has changed its paradigm and becoming more towards a metagenomic approach. So I will be showing you that. As you can see, in 1676, first Leeuwenhoek identified bacteria, and then in Robert Koch in 1888, using potato slices, first able to uh, uh, culture the microbes on solid cultures. Then with the development of Sanger sequencing and PCR technique by Kary Mullis, and the sequencing and genomic approach was done. And from 1998, the first 16S rRNA library was prepared in 1990, and from 1998, along with the advent of NGS technology, see how it has been increasing. The microbiology and metagenomics has gained this momentum. So from 1998 to 2015, we have been able to uh, do microbiome project, and have also isolated samples from Sargasso Sea project. So these are the pioneering achievements in metagenomics. So why, as you might be associated now with the term metagenomics, so there is a dilemma whether we should call it as a megagenomics or a metagenomics. Uh, See, this metagenomics is very young in the field and had hardly scratched the surface of microbial technology so far. And even in this infancy, two features are extremely evident. First one is that we have to isolate bacteria or microbes from any environmental samples, and then it will be and send it for sequencing, and it will generate a huge amount of data. So another important feature is that it generates a huge amount of data. Consider human genome, which took about 10 years from 1990 to 2000, and another 3 years. So almost 13 years it took to sequence our entire genome. And even after that, we don't even know certain regions, how they are functioning. So uh, the entire human genome is about 3 gigabases long. And if we consider a soil sample, as I have told you, there would be about 40,000 microbes present. So just think about the amount of data it will be generated. And for a person manually doing and assembling this genetic material will be next to impossible. So here we need a computational biologist, a bioinformatician who will do the task for you, or even if you are interested, you can learn this technique and analyze the data. So metagenomics is becoming more and more synonymous with the term megagenomics, which is genomics which is performed in a larger scale. So here I will discuss some of the important projects which are going on nowadays. So first one will be Sargasso Sea Metagenomics Survey. So this is a sea which is present near Caribbean Sea and Atlantic Ocean. And it is surrounded by streams from 4 sides. So it forms a gyre. And as you can see in this image, it is surrounded by, I'm sorry. So it's surrounded by some brown seaweed, or what we call it Sargassum Sea, where it gained its name Sargasso Sea. So the water remains mostly calm, and from there, the Craig Venters group have isolated bacteria and viruses and send it for sequencing. And through that, survey, metagenomic survey, they have been able to identify about 12 lakh protein coding genes. So you can imagine this 12 lakh protein coding gene was about 10 times of the known protein coding genes curated before that time. So it provides a huge amount of knowledge to us, and we can use it in future. The next important thing is a soil resistome project. So we know that antibiotics, what we use in our clinics, have mostly developed from the soil bacteria. So it will be evident that the antibiotic resistant genes have also rose there. And we know that the bacteria often have an horizontal transfer of uh, antibiotic resistance gene between two organisms. So, and as we know that majority of the bacteria cannot be cultured. So through a metagenomic approach, this will be very important to identify any antibiotic resistance genes which are present there. As one of I can mention as an example, from the Alaskan forest soil, acetyl transferase gene was identified through this soil resistome project. So next one is acid mine drainage project. So the acid or metal mines, or coal mines which are present in different sites, what they do in through the natural weathering processes, so they emit uh, acidic discharges which ultimately merges with the aquatic environment, and as you can imagine, it will be very detrimental for the riverine or ocean stream where these acid products are going. It can kill the entire fauna flora present there. And the microbes are also present even in this harsh environmental condition, and they can survive. And often they take part in this weathering process through nitrogen fixation, sulfur oxidation, iron oxidation. They can facilitate this project. So what is the utility of studying acid mine drainage project is that by studying this microbiome, we will be able to identify the bacteria and understand the metabolic processes like sulfur oxidation, iron oxidation, nitrogen fixation in a better way through this metagenomic process. And maybe we can, by targeting some of the bacteria, we can retard the process of acid mine drainage in that field. The gut microbes, human microbes, microbiome project is one of the important projects is going on. And the bacterias are present, or we are actually immersed in the bacterial ocean. So it is present on our skin, oral mucosa, gut, genitalia. So if being present in different anatomical locations, it can influence our health. It is not only with the disease, but when we ingest any food, it can remove the toxin from it. It can help to metabolize the complex polysaccharides, and absorption of vitamins and minerals will be facilitated by the presence of bacteria. So those who have gone recently to the clinicians with any uh, problem with their gut system might have experienced that doctors don't prescribe antibiotics for the treatment of any stomach problem. They often prefer to eat you probiotics for the replacement therapy of the microbes which might be absent at that particular condition. So human microbiome is very important for us, and in future it will revolutionize the medical sciences. And of course the viral metagenomic project. So this is very new to this field. We haven't explored it very far. We have though NGS technologies available to us where we can isolate the viral genetic material and send it for sequencing. Though we have identified some of the bacteriophages which is present in the Sargasso Sea project, But doing a virome study as a whole, we haven't approached that far because of the absence of any universal gene for its survey. But as we have 16S rRNA for a general survey of the microbial communities which might be present in a marine community or a soil community, but we don't have such a universal marker available for viral for studying viral metagenomics. Once it is available to us, the metagenomics will gain momentum and will advent in future. So we can classify metagenomics into two fundamental things. First one is sequence-based metagenomics, and another is a functional metagenomics. So if we have an idea that we only want to know who are they or what are the things are present in a particular environment, we can go for a sequence-based metagenomics. We don't need to analyze its function. Maybe it will be a later approach, but mostly to studying biodiversity or relative abundance, we can go for a sequence-based metagenomics. But for the functional metagenomics, if we want to screen what are the things they perform, say for example, what are the things which are associated with sulfur oxidation, what are the genetic signatures which are present with nitrogen metabolism, then these kind of approaches will be it's mostly an experimental one, so will be a functional metagenomics. So through this functional metagenomic approach, we will be able to identify novel protein coding genes, enzyme which might be important to the industries. So in details, as you can see here, that a sequence-based metagenomic study can be used to assemble genomes, identify gene and complete metabolic pathways, compare organisms in different communities. Say, for example, you have isolated bacteria or have performed a sequence-based metagenomics from site A, and you are comparing with site B, then it will be a comparison of their genomic signatures, which particular bacteria are present or absent in that environment or community. Or we can, depending on the environmental conditions, we can say this the relative abundance or correlate the relative abundance of any particular bacteria in it. It also can lead to a better understanding of how certain genes which are present can survive, help us to survive in a particular environment. Sequence-based metagenomics also can be used to establish a degree of microbial diversity and the number of bacterial species in a particular sample. And of course, analyzing microbial diversity can provide valuable information about the ecology. As I have told you that a bacteria, whatever smallest proportion it might be present in a community, it might be doing a very important job in that environment. So removing that particular strain or removing that particular bacteria or microbe from that region might be detrimental for its ecology. So if we want to study any ecology of microbes, it is important to study it as an entire community. So functional metagenomics, it's an experimental approach for extracting the DNA or mixed microbial population, and information is obtained from functional genomics can help us annotate future gene functions and serve as a complement to the sequence-based metagenomics. Functional metagenomics allows us to discovery of novel enzymes whose functions cannot be predicted from the DNA sequences alone. So what we generally do in our genomic approaches, when we have a sequence, we try to translate it into the protein, and through some bioinformatic analysis, we can predict the whatever structure and function it can have. And from that, we can predict what novel enzymatic functions it can do. But functional metagenomics directly provide you an opportunity to identify novel enzymes by culturing it, those who are able to provide that enzyme and directly sequence those genome and identify that gene. So the classical way of doing metagenomics is that we fragment the genetic material as we have done for human genome project. So what we, for the human genome project, what we have done, we have fragmented the human genome, and taking a small part, we have cloned it into bacterial plasmid or phosmid or cosmid, depending on the amount of insert it can intake. And then we have transformed into bacterial cells and grown that bacteria to form a particular colony, and then isolating the genetic material and send it for sequencing. For the classic metagenomic approach, we can also do that. We have we clone the genetic fragment of the isolated from the environment into the plasmid and grow them and then send it for sequencing. This here is a blueprint of whatever metagenomics study you want, what are the things you can want to perform. So if you have an idea, if you are very new to this metagenomic field, if you have an idea that you only want to make a survey, what are the bacterial community or what are the biodiversity which are present in an environment, you can go for a sequence-based approach. You can screen 16S rRNA from the bacterial strains and then identify what are the relative amount of bacteria are present, or what are the bacterial cells are present in that region. So that will serve the purpose. So for doing that kind of metagenomics, we need to select a site where you want to perform a metagenome. Then from the collecting the environmental samples, directly isolating the genetic material that may be DNA or RNA, and then send it for sequencing. And after that, the sequencing through, if you are performing NGS study, will get a huge amount of data. So you have to assemble those small fragments and annotate the gene functions and compare the complete genome to a available reference database. So that this is what how you perform a metagenomic sequence-based study. But if you want to know what kind of functions it is doing or how it is doing, so then you need to do the same thing. You have to select the site, isolate or recover the genetic material from the site, create a library, and go it for and assessing its function. So that's why you will be able to know what are the things they are doing in that field. So these are the basic steps. I will be briefly explaining it today, and in the later phases, next days, I will be going into the details of how we perform metagenomics in a wet lab. So the DNAs are actually fragmented in small pieces, and then it cloned into plasmid vectors, or you can, if you are performing a 16S RNA, you don't need to do this. You only need to perform 16S rRNA amplification and send those amplicons to next gen sequencing. So so these kind of things can be done, and once the sequence is available to us, we can use a computational biology for assembling the genetic material and then send it for annotation and assigning the gene functions. So for the isolation of the genetic material, as I know that you have to directly extract the genetic material from the site. So there are three available DNA extraction techniques you can go through. First one is organic extraction, where you use a phenol chloroform based methods, where the lipid and proteins are dissolved and leaving behind the genetic material from the cells and in an aqueous phase, and you can isolate it and use it for your subsequent downstream analysis. And there are other two techniques which are available, and there are many companies which provide you as a kit, where you can directly go for the magnetic bead based separation, where we have paramagnetic beads. And if you want to isolate the genetic material, these paramagnetic beads are either coated with carbon compound or silica based compounds, and in the internal material is a paramagnetic bead. That means it shows paramagnetic property when it is exposed to a magnetic field. So what we do, we mix these magnetic beads along with the lysed sample and apply those genetic material along with the magnet bound to it. And when, as you can see here, wash the genetic material will be isolated from the debris, and you can wash it and remove the excess debris material from it. And when you elute it, the magnetic bead will be separated from the isolated genetic material. So you can elute the DNA and use it for your analysis. So next one will be a spin column based analysis, where we have a silica based column, where the homogenized tissue is passed through centrifugation process, and only the genetic material will be left binding with the silica material, and the other things which are lysed are not a part of the genetic material will be washed away. And once you elute it, you will get a pure genetic material and use it for analysis. So as I have mentioned several times, the use of 16S rRNA. So you know that 16S rRNA is a component of ribosome, and rRNA are the most abundant type of RNA which are present in a cell. So and there is slight difference between a ribosome which is present from the prokaryote with the eukaryotes. So the largest subunit contains 23S rRNA along with a 5S rRNA and forming a 50S subunit. And the small subunit, which is 30S in size, have 16S rRNA, as you can see here. So this 16S rRNA is our prime target. It has several variable regions and hypervariable regions which have diverged over time, and several conserved regions which are also present. So using those conserved region, we can design primers, and through PCR technique, we can amplify those regions and make a survey of the genetic materials which has been isolated from different sites. And this will give you an idea by analyzing the variations which are present in those hypervariable regions, what type of particular bacteria might be present there. And we can make a phylogenetic tree or a biodiversity map based on that. So this is a sequence-based approach where we use 16S rRNA. Where we don't need to identify any other gene functions. We just only need to know the function or what are the things are present in that soil, or what are maybe any genetic material. So these are there are several advantages and disadvantages of using 16S rRNA. So it is universally distributed, as you know that majority of the bacterial archaeal strains have 16S rRNA. So using 16S rRNA as a general approach, we can identify the bacterial species and strains. And it's very easy to make a phylogenetic relationship across the taxa. And performing 16S RNA is very cheap compared to the whole genome shotgun sequencing. It's all about only $40 to $60 per sample. And of course, there are disadvantages also because of the high copy number variations. We might end up identifying a bacterial strain into a different species or ambiguous genetic structures. So the relative abundance cannot be estimated because the relative abundance cannot be, if you are trying to do an 16S rRNA sequencing analysis, you know that it is based on amplifications, and a typical eukaryote cell can yield up to 6 picogram of DNA from one cell. And if a particular soil or any environmental sample have a very low count of microbes in it, the amount of DNA will be extracted from that species or genus will be very low. And maybe it will not be sufficient to perform a PCR reaction. We at least need 50 nanogram of DNA to perform a successful PCR reaction. So as this is based on amplification, so there will be an amplification bias, because only those will be amplified which have relatively high abundance in that isolated genetic material. So if you are trying to do a relative abundance study, 16S rRNA is not a very good idea. So diversity of the genes tends to be overinflate the diversity estimate. So you will be rather there will be no limited variations present, and you will be estimating that there are many numbers, and this anomaly might be created if you use 16S rRNA. But as the sequencing costs are getting reduced day by day, we are moving to whole genome shotgun metagenomic approach rather than performing a 16S rRNA for our study. Now, one thing you will repeatedly observe in metagenomics, that is coverage and read length. If you are doing a study from NGS, and you know that in several NGS techniques, they yield different read lengths along with different coverage. So if you are isolating the environmental samples, there will be a large distribution of species, and each species would have accounted for individually. You would need to know the number of total species in that sample, the genome size and relative abundance for such species. In most cases, this is not possible when you are sequencing for a sample for the first time, because you don't have an idea what could be present in that thing. So for a coverage means the amount, the time, number of time you read a particular base present in a particular position. So if, for example, if you are reading a base 50 times, that will give you a 50x coverage depth for de novo assembly. So if you have 10 bacterial species estimated with a genome size of 2 MB, you would gain an aim for about 2 GB of DNA sequencing data. So 10 dominant is a very easy calculation. So a dominant bacterial species with 100x of coverage, each with 2 MB of genome size, will give you a 2 GB of data, approximately comparable to human genome size. So what are the applications we can use for metagenomics? For the identification of novel therapeutic compounds, industrial enzymes, clinical diagnosis of the disease. Say, for example, if you compare a diseased person along with a healthy person, and if you identify the presence or absence or relative expression of any particular bacteria or microbe in any of the condition, you can correlate the disease status. It can be useful as a biomarker for that condition. Even the presence of such, like I can mention, the presence of oral microbiota can be an indication of future oral carcinoma, or the bacterial strains present in a genitalia of a female genitalia can be an indication of cervical cancer. So the pathogen monitoring in the environment. Say you are environmentalist. You are just surveying the riverine streams and trying to identify whether the industrial areas are releasing any toxic effluents from its site into the riverine stream. So a very good way to do that will be monitoring the microbiota from different sites of the riverine stream and identify the presence and absence of any particular pathogen or any particular microbe which might be an indication of what kind of or where the toxin has been released. For the agricultural development, if you are associated with agriculture development, you will be very much interested to know what kind of bacteria present in the soil can bring more fertility, will be essential for providing sulfate and micronutrients to the developing crops. You can analyze or monitor the pollutants which are present in the environment because of the presence of any particular bacteria. And of course, as I have told you, for the bioremediation, for the removal of toxins which has been present in the environment. So for the human microbiome, we have all over the place about 100 trillion bacterial cells are present in different anatomical locations: nose, mouth, lungs, stomach, colon, sexual organs, skin. And the presence of this microbial population can be influenced by genetics, environment, or whatever we eat, our lifestyle, and even hormones, and even industrial or even industry. So this is becoming increasingly important. Those who are interested in pursuing their career in uh, uh, human health and medicine, they can easily study these in future and pursue their career. So as I was talking about this human microbiome, so if you go through the current literatures, you will find that there are becoming more and more important. So one of the future medicine, it might seem you as a sci-fi movie, that a poop transfer can treat you a disease. Can you imagine just a small fecal matter from isolated from any healthy individual and transplanting into any diseased individual might cure your ability. And it has the presence of the microbiome in your gut have a profound influence on your personality or your mood or even neurological conditions. Not only in neurological conditions like type 2 diabetes or type 1 diabetes, instead of treating with insulin, you can just replace the microbiota and have a complete cure of that disease. So for the type 1 diabetes, they are proposing that this is might be the actual things might be happening. So as I have told you that the microbes which are present in our gut, they constantly interact with the immune system. So our immune system, which is an adaptive immune system, they eventually learn how to interact with the resident microbes, commensal microbes which are present within us, and thereby, and on a certain condition, if a particular bacteria is removed from the system, maybe due to any particular condition, so in that situation, those cells might become hyperactive or reactive to our own organ. Maybe that might be our islet of Langerhans, which might be producing insulin. So thereby you might be developing type 1 diabetes. Even our obesity and skin diseases and cancer, these things has been associated with microbial populations which are present on Earth. So you can just imagine how it is important for us to study microbes. Even microbes present on us can be different to one person to another. So acting as a fingerprint, the presence of any particular microbe, say for example, you are studying any dead person in a post-mortem study, the presence of bacteria on the surface or its intestine would be an indication when that particular person has died, or it has been toxic, has been applied any toxins or not. So using it can be extremely useful for forensic studies also. So along with the profound benefits of metagenomic study, of course, there are certain limitations are also present. So first one is entry level access. It's still inexpensive. Still, if you are trying to plan whole genome studies using NGS, you require a certain amount of money to take that opportunity. So a comprehensive ness of the genome catalog. So we don't have all the reference genomes available. So even if we identify any gene, we don't have any reference gene available to, or closely related gene available to make an assembly. So we have to rely to some extent on the available genomes, which might not be very exact, but using that we can identify the sequences. Biases in microbial or the functional profiling. So as we know that we don't even know the proper culturing conditions, so to make a functional profiling, it's still a drawback for us. Microbial dark matter, which is a very important. Even as we in our genome, there are certain regions where we don't have any idea what are the unknown regions are there. So for human genomes, similarly for the bacterial genomes, there will be certain regions available which will be remaining as gaps. So we don't know what functions or what importance it has in maintaining the microbial genomic integrity. Culturing microbes directly from the environment, you will not have any idea whether those bacteria are dead or alive. I would like to give you an example. As you are all going through this COVID pandemic, we apply uh, an RT-PCR technique for identifying viruses, the presence of viruses. No technique has been identified yet where we can isolate the RNA and tell whether that virus has or that RNA has been extracted from a dead virus or a live virus. Similarly, for the microbes, we don't have any idea until we perform a functional analysis whether it has came from a live or dead bacterial population. So the curse of compositionality, the quantitative metabolic metagenomic features are reported as a fractional values without links to the real absolute concentration. So the variation in the true concentration of the organism across the samples thus produce false corrections. So even if you are identified a novel change, you might not be very sure whether it has being present there or it came as an anomaly. So for example, a highly abundant organism doubles its concentration in two otherwise identical samples. All other organisms present in the sample will be differentially abundant after normalization. And mucosa associated microbiome, not only mucosa associated microbiome, if you are isolating microbes which are present on any particular host, it might be plant, it might be any human or animals, along with the microbial population, there will be huge amount of host DNA coming with it. So you don't have any choice of eliminating that. So you have to perform the sequencing studies, whatever you are doing, shotgun sequencing, and then through your genetic analysis or post analysis, you can eliminate the human associated genetic features and isolate your microbacterial genome and assemble it. So the future, talking about the futures, it's of course it's a new thing. It's very nascent, and with time it will progress, and of course it will become integrate part of the research activity what we perform nowadays. So to identify new enzymes, antibiotics, of course metagenomics will be the future. So assess the age, diet, pathogenic state, inflammatory bowel disease, Crohn's disease, obesity, cancer, distal gut microbiomes living on humans of different samples will be important. A study of more exotic habitats will be important for us. And extreme conditions like hydrothermal vents and other unexplored regions on Earth, study of antibiotic resistance genes through soil resistome project. Bioinformatics will quicken the analysis of the library profiling. So we are mostly biologists, so we don't know how to analyze this big amount of data. So here is an opportunity. You can collaborate in future with any bioinformatician or computational biologists who will be interestingly analyze your data, and you will be doing the wet lab studies. So discoveries such as phylogenetic tags like rRNA genes give momentum with the growing field. So as I have told you, for the Virome project, we don't have any idea of how we can grow for the analysis. So if you are interested to identifying any universal marker for that, you are welcome. So the learning novel metabolic pathways or novel pathways would lead to understand of the non-culturable bacteria. So yeah, as we don't know how to culture them, directly sequencing will be a good idea for through shotgun or whole genome sequencing. So that's all for today's talk, what we have discussed today.
Thank you, Mr. Shubhrajit Ray.
Thank you.
If there is any question, I cannot see any question here. Just a simple question from my side.
Sure.
We are using metagenomics for mainly bacterial study, bulk amount of bacterial study. How far it is applicable for, yes, I know it's applicable, but how far it is suitable for bulk study of bulk sequencing of fungal DNA?
As far as it is the marker genes are available, it is applicable for fungal genes. Well, though no such studies has been performed in comprehensive studies has been performed yet. There are 1 or 2 publications you can find where they have performed it, but until or unless you are very sure of identifying any universal marker for the fungal community, it could be hard for analysis, but you can do it. So if you go for 18S rRNA or internal transcribed sequence, you can identify the bacterial, the fungal strains present there.
Okay, and during the analysis of gut microbiome, not microbiome, gut, actually we are going to analyze the DNA sequences from the organisms present in the gut. So we can find the DNA sequences from the parasites also, actually who are the eukaryotes. So how could I differentiate between this?
Yes. So as I have told you, when you are
Some are protozoans, some are nematodes, and some are bacteria itself. So how could I differentiate?
Yes. So when you are isolating any, that's the problem. So rather, I would not say it's a problem, but because it will provide you an opportunity to learn about other species too. So when you are isolating all the organisms from the gut, say for example, you are using fecal matter from any individual. So you will be isolating nematodes, protists, and all of them, even the human DNA samples will be available in that one. So after this generation of the sequence data, so you have to imply along with them, because you know the C for the C elegans, it's a nematode. So you will have the reference genome available, so you can identify what are the things present which is similar to that thing, and you can isolate from that entire pool and then go for identification of other things. So whatever will be left, you can go for de novo assembly for identifying the bacterial species. So the hardest part only will be for the unknown one. So whatever the things will be available to us, you can the final tier where we will be identify the unknown bacterial or population. So you need to know a de novo assembly. Then only you can go for identification of that phyla. Otherwise, most of the things what we are doing are clinically relevant to us. Their reference genomes is available, and you can compare it and identify it.
Okay. Any question from anyone? No question. So, Dr. Shaurajit Dash. Shaurajit, Dr. Shaurajit Dash. Hello, Dr. Shaurajit Dash.
Sarojit Das, sir, are you there? Sarojit
Yes.
Das, sir. I think so, ma'am. There is some internet connectivity issue.
Okay. Okay. So in that case, if there is no question, I will thank you again, Mr. Shubhrajit Roy, for his enchanting discourse, and we hope to hear and to learn a lot on this metagenomic studies and the next gen sequencing in the coming days. So with that, I would like to end this session. Thank you.
Thank you.
Thank you. Prem Kumar, can we end the session? Hello?
Yes, ma'am. Yes, ma'am.
So please.
So there
Thank
is small instruction for all participants that we will be back tomorrow at 9:30 a.m. and all the links will be sent to you via mail. So the day 2 session will be at 9:30 tomorrow. So we will be back tomorrow.
And please put your questions answer in the question answer box or in chat box. Please don't forget to do that. Okay, thank you. Thank you again. Thank you, Prem Kumar. Okay.
Thank you all participants. Thank you all.